The cell cycle is the life cycle of the cell, and this involves taking a single cell, growing it, and then ultimately splitting that into two. This involves two major parts. We have interphase, which is going to be the growth part, so that's the first portion of it, and then we have the actual splitting portion, which is known as cell division. And cell division, that's the reproduction of daughter cells, so getting two cells from the initial parent cell that we started with. Now, this process, it does happen pretty regularly with our cells. So why do cells divide? Why do they replicate? Why does this need to take place? Well, it is very important for our overall growth of our body. If we are talking about growing, that growing involves producing new cells. So we have to produce new cells to grow our body, grow our organs. This is also important for tissue replacement or tissue renewal. So as we have cells that die off or are damaged, we must replace those. And to replace those, we have to produce new ones. A final reason would be just for the reproduction process. Just for reproduction, we will have to have new cells produced so that we can get new individuals, new organisms. So as we talk about the two parts of the cell cycle, interphase is the first portion, and that is the growth process. And so in this image, we can see that we start with the cell. It does get bigger to emphasize the growth. Interphase is the longest portion, the most time-consuming portion of the cell cycle. With interphase, just to sum up what we're doing in interphase, is we are preparing for the second part. So it's a preparation phase. And with that preparation phase, we have to copy the contents of that cell. And that's why we see it getting bigger. What we're most interested in, most focused on copying during interphase is going to be the genetic information. So interphase is going to require careful duplication, careful copying of all of the DNA inside of that cell. We're focused really on the nucleus because the nucleus is where the majority of the DNA is located at. So to look at the DNA is a double helix. And that double helix is going to be carefully peeled apart from each other, and we can kind of see that here. It's going to be peeled open, and then we will actually copy each half of that so that ultimately we do end up with two double helixes. If we were to draw that, we would basically start with something like this, and then that is going to get turned in to two molecules. So we'll have two of them from the initial one that we started with. So we're taking one and we're making two copies. After we get that done, then we can actually take what we have in those two copies and split them apart into the two daughter cells. Because these cells that we end up with, we want them to ultimately be identical to each other, and we also want them to be identical to the original parent cell that we started with. Mitosis is what we call the splitting process. This is going to be careful division of the nucleus and everything that's inside the nucleus. And so what that means is that we are carefully dividing up the DNA at this point, making sure we have one copy of everything here and one copy of everything over here on the other side. Two daughter cells is what we will get at the end. Just to emphasize here, this is what we call the parent cell. These over here are the daughter cells. And so these daughter cells, they are supposed to be the same as each other, but they're also supposed to be the same as this one that we started with that we're actually splitting in half. Mitosis does get divided into four phases. We have a prophase, metaphase, anaphase, and telophase. Along with telophase comes the actual splitting of the cell, and we refer to that as cytokinesis. All of these four phases together are extremely short. Interphase is the longest by far. Most of the cell cycle, the whole thing takes place in about 24 hours. And of that 24 hours, about 23 hours is for interphase. And then these four phases are all buried into 1 hour. So this really is a fast process when we talk about actually doing the splitting. If we look at the actual splitting quickly, we see all of the phases here on this slide. We're starting with interphase. That is that preparation phase. And then ultimately what will happen is we have to get rid of the nuclear envelope or the container that's holding everything in there. At the same time, the chromosomes will just condense and compact so that they will be easier to separate. Notice we have some strings that are forming inside. Those are part of the cytoskeleton, and they will help to move our chromosomes around. They will attach to the chromosomes themselves in prophase, and then we will see that they get carefully lined up down the middle. That's going to be our metaphase. Once they're lined up along the middle, in anaphase, we actually pull them apart and we see things moving in the two directions. Same things are moving to this side compared to the other side. And then in telophase, we basically repackage those two groups. So we build a nuclear envelope around this group and a nuclear envelope around this group. And we also split that overall cell into two, so that at the end we have two cells, daughter cells, that are identical copies of each other. They each have their own set of genetic information as well, and that is an exact copy of the cell that they started with. So the cell cycle is what our bodies use when they need to produce new cells. And this would be new cells because we are growing. This could be new cells because just some of them got old. It could be new cells because some got damaged. For example, if we have an injury on our arm, like a cut, we don't tend to worry about that too much because we know that that will heal. Well, when we use the word heal, what is really happening there is that the surrounding cells, the ones surrounding that injury, they are going to divide, produce new cells to replace the ones that just died there on our arm. So we do heal with the production of new cells. Protein synthesis is going to be very important for just overall running our cells. DNA is the blueprint that our cells use, and what we do specifically with that blueprint is that we read it to look at how we want to make our proteins, or how we are going to make our proteins. When we talk about genes in the DNA, that is what the gene is. The gene is information to make a protein. Our proteins are made of amino acids, but they have to be assembled in the right order. So why we need proteins? Lots of different reasons there. Proteins are the workers inside of our cells. They are running all of our different activities. And if you can just think of a function that the cell needs to perform, there's going to be a protein that does that. Some of our proteins are enzymes. We've talked about how they are involved in transporting things across a membrane. We have proteins involved in the cytoskeleton. There are hormones. So just long list of functions that our proteins can actually perform, and that's why we will be producing so many of them all the time. To get the proteins produced, though, there are several steps involved. So to do that, our DNA is sitting in the nucleus, and the first thing that our cells have to do is they have to take the instructions to make a protein, and they have to first copy it into RNA. And this will be done inside of the nucleus, and that process of taking the DNA and copying a section into RNA, that is known as transcription. So we are transcribing, or making a tiny little copy of the DNA, and once we have that tiny copy, that copy is going to be sent out of the nucleus. When it exits the nucleus, that would be through one of those nuclear pores. At that point, that little copy is now in the cytoplasm, and we can read that copy and turn it into a protein. So we would then have our protein factories, or the part of the cell that's responsible for making proteins. It would be looking at that. So specifically, the ribosome is going to look at that little copy of RNA, and it's going to understand what it means, and it's going to turn that into a protein. And that process of copying the RNA into a protein, that is known as translation. So that is specifically what the ribosome is doing in the cytoplasm of the cell. And at the end, we have a protein that is now going to be able to function inside the cell. So anytime our cells need to make protein, the first thing that has to happen is we have to go to this massive quantity of DNA that's in the nucleus, find the section that has the instructions for the protein we want to make, copy just that little chunk, send it out to the cytoplasm, and then the ribosome will use those directions to make the proteins that we need. In that way, the nucleus is kind of controlling and having the information to make everything that we do ultimately make inside of the cell.